Okay, that's looking good. Yeah, good day. Thanks for having me. And welcome to my presentation about microalgae in research and application. My name is Christina KUCHENDORF, and I want to give an impression today about semi-industrial algae production at the Research Center Jülich in the Northwest European region with a few different production techniques. I just want to give a ballpark estimate about what kind of productivity can be expected, but also what needs to be taken into account for bioeconomic calculations when planning your work with microalgae. I myself am from the Institute of Bio and Geosciences, the IBG2, at the Forschungszentrum Jülich, where we set focus on more efficient plant production on several levels, from the understanding and improvement of plant or algae, in this case, performance itself, from laboratory to greenhouse to outdoor, including the scale-up. And this goes to the methods to track it and improve on it in greenhouse and field with the target of a sustainable bioeconomy. So why have microalgae become interesting to us? So most of us know already what can happen in a neglected fish tank and how fast this can happen. But how to harness these qualities? So the possible green resource microalgae, as hinted in that fish tank, has a huge growth potential, as we've already heard, but only very few species are already being exploited. So there's still a huge potential to tap into, especially since even closely related species can have hugely different traits, like different growth preferences, like the high temperature growing chlorella. There are chlorellas which like high temperature, some which like low temperature. They're just so different inside this one genus. Or for example, the lower temperature thriving Scannidesmus we have in our winter. There is a salt preference likers like spirulina, or those with certain already very variable pigments like the red Haematococcus. So in this presentation, I want to focus on the phototrophic microalgae production. And right now you can see a few pictures of algae production sites worldwide and in Germany. You may realize that the German production sites so far, these are commercial ones, they're all indoor set. So just some examples about the usages of microalgae today. So many species, or many growers, maybe one should say, prefer saline conditions because it's a more stable production. But in turn, it may complicate the handling of water use or reuse if you, for example, put it in combination with agriculture. But several species are also or only very productive freshwater species. And some products like the pharma or nutraceuticals need to be heavily processed, while wastewater treatment or nutrient cycling may require the wet biomass. And this also needs to be calculated before starting into an algae production, the often necessary water removal for transport or processing being a very energy and cost-intensive step. For example, an algae culture normally comes at something between 1 or 10% of dry matter in water when you harvest it. It depends on the cultivation type, but you will almost always end up with a huge amount of water you will need to remove. That doesn't depend on if you reuse it or not, but at some point you have to remove some of it. As to the current usage and yield, I've brought some current trade prices of microalgae biomass, which actually they vary a lot if you go to the net or to the market and check. But this is kind of a ballpark for this. So expensive products, of course, help establish larger production sites. It hugely helps with the investments and the reasoning of it. And the same is being said for the usage of all available synergies. For example, like we do in Jülich, we use the waste heat and the CO2 from a lignite plant next door. Or it would also be possible to couple it to a greenhouse wastewater or something like this. The efficiency is very highly dependent on this, and of course also on the targeted product, like also Benham just said. And very much too, in case of the phototrophic production, on the location. So on this list, you can see that Jülich only had a 4-ton production before 2017 per year. This was a time when we were concentrating on oil-rich biomass. And this changed later when we were able to take better into account which organisms actually does well in our conditions and take into account the local prerequisites we have, as we will see in a few sets later. So it's really about location, 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 and making the right decisions. So here are some images from possible products from algae biomass. Some of you may have seen part of it before. There's pigments like used in wine gum, for example, from food additives and feed, but there's also the possible usage as water cleaners. So here's an example coupled to our local wastewater treatment plant. I will get back to that later again. And biomass from this can be, for example, used as a plant fertilizer, which we have also tried and published, which actually worked surprisingly well. So for this latter work and all our previous and current, we use the facilities of the so-called Algae Science Center in Jülich, where we have 3 times 500 square meter for experimentation in and outdoors. Currently, because of COVID-19 and personnel issues, we have reduced to experimental work on much smaller scale. But in general, the maximum capacity of these facilities combined was between 65 and 70 cubic meters of algae solution. And at the time they were built, all of these 3 facilities were commercially available. So because most of them, at least the foil greenhouse and the outdoor facility, have been just one large circulation of algae, we have reversed now to make many, many smaller segments of this to actually be able to experiment more with this. But this can, of course, also be reversed. But it makes it easier for scientific work to have segmentation instead of the huge bulk production. So a bit closer to the systems, there is the relatively cheap but work-intensive so-called Vback system within the greenhouse. All of these bags you can see are interconnected, so that is actually one cultivation you see here. That's a lot of connections. They are going like 4 tubes into each of these bags. And it can these bags have a huge advantage that they can, of course, also be used single or for tests or starter cultures for this or for the other facilities. But it needs a lot hand-on work. Then there is the a so-called spray technique in a mesh ultra-thin layer bioreactor. Which overcomes the problem of having bubbles pass through your culture for gas exchange with an air CO2 mixture. And the bubbles normally tend to pass very quickly and don't leave much time for CO2 to actually dissolve in the water. So here the concept has been turned around because the billions of droplets which are formed by spraying and dropping through these nets form a huge surface for gas exchange, and the atmosphere inside this greenhouse is CO2 enriched. This works nicely and also with higher algae densities and a high grade of automation, but is very expensive and needs quite a lot of cleaning in between. Then we have the outdoor production side, which still allows some degree of temperature control around the year. So we normally run it just avoiding freezing or overheating in summer. There is some possibility of doing a water heat exchange in there. And it has relatively simple handling and harvesting and a high light exposure, but of course also a high weather exposure. So there are always the advantages and drawbacks of these systems. So it always depends on what you are actually going to use it for. Also, if you now take a view at this huge facility, that's like 28 cubic meters of algae solution. This is one circulation. You do not want to have a leakage in there. You want to have smaller segments in this. So, here I will give you now an overview about the cultivation process. As mentioned, we are now working with a consortium, and actually we are working with a rather old consortium, which is already several years old and is always being reused because the algae in there, the bacteria in there, they know each other. We just take them from outside and restart the cultivation. But if you would want to start from laboratory culture, because you can buy them, it would look like this. So start from a petri dish, you go into a flask. If they have grown into a flask in the nutrient solution, you just continue diluting them with nutrient solution until you can, for example, start such a Vback volume and have them grow in more volume. And this can be harvested continuously, or, for example, when targeting an oil-rich biomass in batches. For example, for this you would need some nutrient dilution, nutrient depletion, which is necessary to push the algae into lipid accumulation in this case. The biomass can be harvested then by means of filtering or, like we did, centrifugation. Many other methods are possible. It depends on the size and cost efficiency ratio of your setup. So for us, we had a limited amount. Even though it is a large production, it is still, in terms of large production, a limited number. So a centrifuge for us was the compromise between how many working hours we need and how many cubic meters we have to pass through the centrifuge. So there are a lot cheaper methods, but this is a question which will always pop up. You have to find the right one for your size, volume, input, output balance. Then, of course, afterwards, you have to find some way to either store it or make it transportable. So for us, we had the methods to either freeze or spray dry the resulting algae paste we had from our harvests. Our results in the recent years from this have indicated that multiple approaches and also lots of compromises are necessary for a successful production in Northwest Europe. So more light, feasible temperatures, and a better understanding of the target algae in this relation is of utmost importance. These are the most limiting factors here. So this is not a trivial task, and not everybody has studied biology just to take care of algae. A simplification of the process and a further automation is needed to increase the accessibility of the technique, so you can put it in the hands of a large large group of users. So it is desirable to have some help in the decision making when caring for a culture, like when to harvest, when to dilute, when to restart, when to feed. And that could be much more easily done with the today available self-learning processes and, for example, connected into the Internet of Things to create an experience database and not repeat the same errors all the time, just in different places. And also the exploitation of all available synergies is necessary, and then algae production will become still cheaper also in Northwest Europe. But due to our very variable weather, it is not yet. I'll also come back to that. In combination with the hopefully growing database, a network between the players in there, the interested people, is very desirable, and intended users of these bioeconomic techniques to obtain sustainability should also heavily focus in there. We are still overall too small. We need to motivate many more players onto the field, and this of course also reduces the cost in the long run. So if you're interested, please join. First steps to this were taken in the project idea. So as you heard, we do not only target the year-round production and the whole possible value chains, but also the interaction with people who are or want to get involved. In this, our group was mostly involved with the stable and reliable cultivation. And this is where I'm coming back to the climate conditions in Northwest Europe, where we still want a reliable year-round algae cultivation. For the other tasks, please check on the project website or on all the partner presentations here today. So as you can see, light and temperature in our area are quite variable. For example, the algae grown in Jülich are very receptive to the difference between minimum and maximum temperature over the day, which in 2020, for example, has been rather large for a long time compared to other years. If you check the red and the blue line in the beginning of the year, so in the on the right side of the diagram, difference between those in the spring was rather large, and this actually hinders the consortium to do the species shift into the summer composition. So this would look like this, because we are working with this algae consortium, which is able to buffer some of the effects of the outer parameters, because the best suited algae in any point of time will be the strongest contributor at this time. All of these algae occur naturally in our area, so they are less susceptible to pathogens, but this also means that we have a seasonal product. So depending on the weather conditions, the biomass consists mostly of one of these different species. If you check here, we have summertime with mostly chlorella. Then when it gets colder, we get a switch to mostly Scannidesmus, and in the winter to the Chlamydomonalis. And then it switches back to Scannidesmus, and normally we would expect in April, when it gets more summery, that it's already switching back to mostly chlorella. But this year, the different temperatures meant that we had to wait until mid-June to actually get back to our almost pure chlorella conditions here. So a culture like this is not necessarily easy to time, but it is very stable in its overall growth. You always have some biomass production, and it doesn't crash because there are always some surviving cells. So the tasks here are, of course, that there has to be some quality management defined of an output you want. The reliability is there, and it can be coupled to several other processes, for example, also to nutrient uptake on waste streams. So we have to get to this question, how can we make now value instead of waste from this? you will be interested in some numbers on productivity from this. And we have experimented with different harvest timings and dilutions and found the consortium to be highly efficient when harvested regularly, like 1 to 2 times a week at concentrations about 1 to 2 grams per liter. So in 2018, we just checked if this thing grows the year round and is reliable. And in 2019, we have pushed it. And this increased the production from these measly 4 tons per hectare and year to over, on average, 30 tons per hectare and year. And this is kind of where it becomes feasible to think further about commercialization. It is, however, easier to produce indoors in winter. Then it is about 30% better within the greenhouse than in the outdoor facility. It's about 15 tons per hectare and year. And it's slightly better outdoors in the summer. Then you have outdoors 80 tons per hectare and indoors about 60 because of the shading of the greenhouse. It's also something to keep in mind regarding the local prerequisites. So there are more steps needed to exploit the feasibility of algae production, especially as food or feed products from non-exenic outdoor cultures also need quality control. And additional uses of the algae growth are also of interest, for example, to help close nutrient cycles. So here, phosphorus or nitrogen. And you can see that, for example, if you use algae to take up these nutrients from wastewater streams, they could be fed back into the nutrient cycle and back to the plants. So just as an example to this, how the cycle can be closed, we have done some experiments in fertilization of barley and wheat by algae biomass, which works rather well if the formulation is right for the substrate. And the biomass to do this can be grown in the shown facilities, but it can also be derived from wastewater. So here's, for example, first, what we did conventional in our systems. You have the wet algae biomass and you have the dry algae biomass, which can be mixed in the soil. And you can also use algae from so-called algae turf scrubber. Here's an example from a very small facility coupled into our actual wastewater treatment plant, which cleans wastewater effluent of the remaining phosphorus and nitrogen. And the biomass can also be used well as a fertilizer. And this is only like 1 square meter area, taking up nutrients from half cubic meter of these wastewater effluent just in a few days. So it was rather efficient, even if it's so small. And then we tested, of course, also if it's really the phosphorus taken up by the algae, which ends up in the plant. So we did it one time in the laboratory with radioactive 33P. And you can see on the small picture on the left that we gave the phosphorus on the soil, and the brighter it is, the more radioactivity. So the plant actually really takes up the phosphorus from the algae. So we said, cool, transfer is successful. We need to go further into this direction. So to follow this even further, it would also be possible to couple algae production into multiple processes, not just in agriculture, but also in urban processes. For example, in rainwater clearing, in gray or yellow water processing, or in room climate, either by CO2 removal or, for example, with algae facades for climatization, etc. An example for this would be the BQ algae house, which you can see in Hamburg, where there are facade reactors which are being used for biomass and biogas production and also coupled into the climatization of the house. So we are today at a point in time where these techniques are all available in some kind on their own, and it's now the task to find couplings of these that work well, not just with algae, but also with, for example, photovoltaics, solar heat, geothermal applications, biogas for home use, etc., etc. So there are good solutions already for the energy side of this. But much of the mass and waste streams, especially for water, freshwater and biomass, are still mostly lost after use by us. And these still contain valuable resources, and we seriously need to improve on the true circulation of these resources. And it doesn't only go for agricultural applications, it also goes for urban applications. And this might be where the connection between people interested in electronic solutions and clever automated systems might actually help to push this forward a bit. Now, to combine these available techniques intelligently, the standard is that we ask to use secondary metabolites as a stepstone to get more installations commercially successful with algae, to be able to make cost calculations with better estimates also in our weather regions, to get to an infrastructure, and we need this communication and networking. That always seems the most important thing to do. And then we actually get a broad spectrum of processes and choice, which need to be made a little bit simpler for a broader public in there. And then we get some very flexible and variable pillars for the bioeconomical circular economy, which we target, with a huge synergistic potential. So for the implementation of this, like wherever possible, to gain more experience and make this more available and accessible, we count on your interest and want to motivate you to take an active role and therefore pave the way for a successful, sustainable future bioeconomy, hopefully with microalgae. So thank you for your attention. And yeah, I think then, Benham, it would be time for questions for the whole session, right?
Thank you, Christina, for